Guys, so we are going to take a look today at the structure of DNA. Hopefully, you have your student note catcher open and we will take it from there. All right, so first thing we want to talk about are our learning objectives for the day. By the end of today and doing these notes, hopefully, you will be able to identify the chemical components of DNA. Number two, you will be describing the clues that help scientists determine the structure of DNA. And finally, you will be able to explain what the double helix model shows about DNA. So, the main idea here, or if we want to talk about the story that we've been talking so far about DNA, is that there was some three original experiments, right? There was the Griffith experiment of transformation, followed by Avery's process of elimination experiment, followed by the Hershey-Chase experiment, which of course allowed DNA to be the definitive molecule of genetic material or genetic inheritance. The thing was, after that, and they knew that DNA had to do three things, right? We knew that it had to store information, it had to be able to copy that information, and of course, it had to be able to transmit that genetic information to the next um, generation. But scientists really didn't know how DNA did any of that. And part of the thing that they had to understand then was to understand the structure of DNA as a key to understanding how genetics and how genes actually work. All right, so let's start with the idea of the nucleotide structure. So DNA is one of our macromolecules. DNA, that Na stands for nucleic acid. It is one of our four main biomolecules. Remember, these are very large organic molecules. There are three other ones besides the nucleic acids. There are carbohydrates, there are lipids, and then there are proteins. And all of them are polymers. Now, remember, for nucleic acids, these are very long strands or chains of polymers. And these polymers, these chains, I should say, are held together by covalent bonds. So these nucleic acids are made up of nucleotides. These nucleic acids are built from smaller units, which are called monomers. The monomer of a nucleic acid is a nucleotide. And each DNA nucleotide is made up of three components: the phosphate group, the nitrogen group, and the deoxyribose sugar. So the phosphate group, for our purposes, will always be represented by like a circle. The sugar is always going to be a pentagon because 5 sugars, pentagon. And then the nitrogen base is sort of this shape off to the side. Now, remember, there are two types of nucleic acid. RNA is the other type of nucleic acid. RNA is the same nucleotide. The only difference being is that RNA has the sugar being ribose, not deoxyribose. So it just has a different sugar on it. All right, so this is our nucleic structure. Again, this top piece here, one circle, one pentagon, one base. That is our one nucleotide. And these nucleotides, again, are held together in these long chains by covalent bond, which forms the backbone of DNA. Now, if we look back at that picture, go flip back, see, I have A, G, C, and T as my nitrogen bases. In this diagram, we have the four nitrogen bases are shown, but actual DNA molecules have millions and millions and billions of nucleotides. If you took all of the DNA inside of your human body and you strung it together end to end, it would be long enough to travel from here on Earth to the sun and back 600 times. Yep, I did Google that. So a lot of DNA inside of you, lots and millions and millions and millions and millions of nucleotides. Nitrogen bases, the four types of nitrogen bases used by DNA are A, G, C, and T. A stands for adenine, G is guanine, C is cytosine, and T is thymine. And once more, they are held together by covalent bonds that exist between the sugar of one nucleotide and the phosphate group of the next nucleotide. Now, these four nucleotides, kind of like our 26-letter alphabet, you have these 4 letters, right? And they can be strung together in many different sequences. So we can create millions of different combinations of genetic code. Much like our 26-letter alphabet can literally make millions of different types of words in hundreds of different types of languages. Our DNA, these 4 nucleotides work the same way. They have infinite combinations of making different genetic code. So let's look at the solving the structure of DNA. So now we know what DNA is made up of. Let's take a quick look in terms to try to figure out the structure of DNA to help explain why DNA is able to do the 3 things that it does so well, which is store information, copy information, and transmit information to the next generation. So Chargaff was one of the instrumental scientists in this. So one thing that puzzled people about DNA was that it had a really funny relationship in the nucleotides. Chargaff discovered that the percentages of adenine and thymine were always almost equal in any sample of DNA. It did not matter if it was a chimpanzee or a bunny rabbit or an oak tree or even just a bacteria. If he took it apart and looked at it, the percentage of adenine and thymine were almost always exactly equal in any sample of DNA that he looked at. This holds true, by the way, for the other 2 nucleotides as well. And this observation led to Chargaff's rule, where we note that adenine and thymine are always in the same percentages, present in the same percentages, and guanine and cytosine are always present in the same percentages. So this leads us to being able to make predictions, which is what all good scientists do. So the question here is, if there is 30% adenine, then how much thymine will there be? Well, let's take a look. And for that, I have to go to my tablet. So give me one moment. Okay, so let's take a quick look, shall we? If there is 30% adenine present, then remember, adenine and thymine always exist in the same amounts. So that means that there is going to be 30% thymine as well. All right, I need to change to a different color. This is making me crazy. So if I have 30% adenine and 30% thymine, remember, percentages always have to add up to 100. So 30 plus 30 is 60. So there's 40% left. What does that mean? Well, if I have 40% of my DNA left, because again, 30% adenine, 30% thymine, 30 plus 30 is 60. 100 minus 60 is 40. There's 40% of my DNA nucleotides left. If I divide that into 2, that means that I have 20% guanine and 20% cytosine. So again, this Chargaff's rule of saying that adenine and thymine are always the same percentage, guanine and cytosine are always the same percentage, will allow me to make predictions regarding the components, the component of my DNA, how much DNA or how many of each nucleotide are present. So let's take a look at the next thing. If I can make it work. Oh, there we go. All right, I have to clear my screen. I really hate that I don't know how to work this yet. All right, let's go back. All right, sorry about that, guys. All right, cool. So again, And this is some data from Shargoff. You can see Streptococcus, yeast, herring, humans, E. coli. If you look at your A's and T's, they're always just about the same amount. C's and G's, always just about the same amount in terms of percentages. So let's take a look at another problem. Any species has 30% adenine. Just whatever, species X has 30% adenine in its DNA. What is the percentage for the other three bases? So if I know that I have 35% adenine, then that tells me that I automatically should know I have 35% thymine, because adenine and thymine, according to Shargoff's rules, are always going to be present in this equal amounts. All right, cool. So let's figure this out. Now I have to say 100 minus my adenine, 35, plus my thymine, which is also 35. So 100 minus 35 minus another 35 is going to be, what is that, 30? That's what's left over. So I'm just going to split that equally between guanine and cytosine, because 30 divided by 2 is 15. Again, Shargoff's rule helps us figure out how many of each type, or the percentage of how many of each nucleotide exists in our DNA. Let me escape. All right, back to my regular computer. All right, so we have already done this one. All right, so nitrogen bases. Again, ladies and gentlemen, let's take a look. Our nitrogen bases, again, adenine, thymine, guanine, cytosine. And again, there are hydrogen bonds here between the steps. So hydrogen bond will bond the A and the T together. Hydrogen bonds will bond the C's and the G's together. And again, I have covalent bonds on my rungs. So between the sugars and the phosphates, I have covalent bonds. Between the nitrogen bases, I have hydrogen bonds. All right, so moving on structurally. So Shargoff gives us Shargoff's rule, which tells us about that, again, the different base pairs always show up in the same percentages. A and T are always the same. C and G are always the same. A few years later, we have Rosalind Franklin. Rosalind Franklin in the early 1950s, she is a British scientist and she begins to study DNA. She's using a technique called X-ray diffraction, which I spelled wrong. First, she takes DNA fibers on a thin glass tube and stretches them out so that most of the strands are parallel. And then next, she takes an X-ray photo and records it on film. So underneath her picture is the famous photo 51. That is the photograph credited with that made Rosalind Franklin famous. The reason why is because we have this X-shaped pattern. And if we have an X-shaped pattern and things that are running parallel to each other, what we now know is that DNA has to be a coil. That is what this tells us. This X-shaped pattern tells us that the DNA is twisted around each other, so it's coiled together. Having a shape that is coiled or twisted around each other more commonly is known as a helix. So that is just another fancy schmancy word for twisted around each other. We just say helix instead. Also, we see these dark spots, and they're always running parallel to each other. Those dark spots indeed are actually the nitrogen bases, and we can see them stacked in very regular intervals near the center of this molecule. So very, very regular, and they are spaced nicely around the center of our molecule. So this was an amazing breakthrough. All right, so a few years later, by the way, Rosalind Franklin is still working. She's still working on her research. Watson and Crick, their team, they're also trying to understand the structure of DNA. They end up building models. They make it out of wire. But the thing is, they build all these amazing models, but it does nothing to actually explain anything about DNA's known properties, which at this point includes Chargaff's rule. Now, in 1953, they were all working out of England. Watson is shown a picture of Franklin's photo 51, and it like clicks, like literally in his biography he said it was like, it was a watershed moment. And it contains clues to specific properties of DNA. The data in Franklin's X-ray pattern let Watson and Crick, allowed them to build a model that actually explains the property of DNA, which we did not have until this point. And in that same exact year, the pair published their breakthrough model of DNA known as the double helix. So what does that mean? So a double helix basically means that you have a ladder, right? So you have the backbone, the sugar phosphate backbone, and in between the ladder, you have the rungs of the ladder, which are the nitrogen bases. And this ladder is then twisted around each other. If we build it like this, it 100% explains Franklin's X-ray pattern, and it explains many of the properties of DNA. This double helix explains Chargaff's rule, and it explains how the two strands are actually held together. So again, the backbone of this twisted ladder, this double helix, are the sugar and the phosphate. Remember, those are held together by covalent bonds. And the rungs of our little twisted ladder are actually the nitrogen bases. And again, those nitrogen bases have to follow Chargaff's rule. So A and T's are always going to show up in the same number. C's and G's are always going to show up in the same number. When something is anti-parallel, what it means is they're running in opposite directions. Okay, so again, another important aspect of the double helix model is that the two DNA strands that are stuck together in the center by the nitrogen bond or hydrogen bonds for the nitrogen bases are running in opposite directions. This is what we call anti-parallel. This specific arrangement is what allows the bases on both strands to become in contact near the middle of that 3D molecule. It also allows for each strand of double helix to carry a sequence of nucleotides. So each side has its own sequence of nucleotides. Again, arranged like letters in a 4-letter alphabet. So these just have hundreds of millions of different combinations, which is where we see a lot of that genetic diversity in nature. Right, so double helix, hydrogen bonding. So first of all, Watson and Crick could not explain what held the bonds or the DNA strands together, but then they discovered that it was hydrogen bonds forming between the nitrogen bases. This provided just enough force to hold the strands together. Now remember, we learned this before, hydrogen bonds are fairly weak. But they are, like I said, just enough force to hold these two sides of this ladder together. Now, I do want you to note that A and T are always bonding together. And they have a double hydrogen bond. Cytosine and guanine always bond together, which makes sense because they have to appear in the same percentages. And between cytosine and guanine, we will always see 3 hydrogen bond. This is what is known as base pairing. It is the near perfect fit of all of our nucleotides. A and T perfectly fit together with these 2 hydrogen bonds. C's and G's, cytosine and guanines, always fit together just about perfectly with 3 hydrogen bonds. And there are our lovely hydrogen bonds in the middle. All right, so again, base pairing. Hydrogen bonds with thymine is always adenine. Hydrogen bonds with cytosine is always guanine. So then the question came up of, okay, cool. So DNA is super important. We need it for life. It has to go through all of this stuff, carries all of our genetic information. Why on earth is the weakest bond that we know of chemically holding this stuff together? Does it make sense that this molecule, which is so important, is held together by super weak bonds? And the answer is yes. It totally makes sense because remember, DNA has to replicate itself. One of the functions of DNA is that it has to copy itself. In order to copy itself, it has to unzip, and it unzips down all of little tiny weak hydrogen bonds. So that totally makes sense. Again, if the two strands were held together by anything stronger, they might be able to unzip, and if they can't unzip, they can't copy themselves, and then DNA loses one of its functions. Because again, the three primary functions of DNA are to store genetic information, copy genetic information, and of course to transmit that genetic information to the next generation. And by the way, we will see in our next section how it is that this ability for these two strands to unzip is incredibly important to DNA's function. At this point, you should have enough information to fill out your student note catchers. I hope this was informative. And as always, if you guys have any issues or questions, please bring them up in class or email or send them to me via chat in class. Peace out.